Welcome back to the lecture series in cell culture technology. So, during the first 3 class, we have dealt with the myriad of opportunities which cell culture technology has offered and will be offering in future. We got a glimpse of it using cell culture as a tool for sensing, as a sensor element, as a bioreactor to produce different kind of compounds of biomedical as well as other significance. Apart from it, we highlighted the fact that understanding the fundamentals of cell culture technology and the basic philosophies will be one of the very basic prerequisite for stem cell biology and regenerative medicine. Apart from it, the dream of mankind of having artificial organs. And when I talk about artificial organ, here we are not, I am not talking about extracorporeal devices where you have dialysis bags or all those other kind of things, or heart lung machine. I am talking about a kidney developed from biological elements very similar to our existing kidney, or a heart developed using cardiomyocytes, endothelial cells, so on and so forth. That is where the future is. Now, while talking about the biology of the cells, we talked about the oxygen tension, oxygen and carbon dioxide. Very briefly, of course, we will come back to these things. Then we talked about extracellular matrix protein, and there we discussed the opportunity of using extracellular matrix system to discover newer and newer extracellular matrix, which may be even synthetic analogs, and how a cancer cell cheats by moving out from its actual extracellular matrix profile and kind of like a rogue cell colonizes at different places. While talking about this, we also talked about the concept of adhering cell and non-adhering cell. So, we have exclusively talked about the adhering cells, but just if you think of it a little bit louder on this. So, you have these non-adhering cells which though start their journey from an adhering environment. Say for example, if we take blood, if you think of the blood cells, it will be something like this. Okay, let us once again, this is our week 1 lecture 4. So, this is the 4th lecture, and talking about non-adhering cells. So, if we take for example, if we take the example of RBC or red blood cell, which is also called erythrocytes. These erythrocytes have their origin in the bone marrow, somewhere out here. From here, these erythrocytes, initially they divide from hematopoietic stem cells and they migrate. And they migrate through the blood vessels, and they have a limited lifespan. Afterward, it gets destroyed. So, one interesting feature about these cells are that they travel all over the body, yet rarely they anchor at any point. So, apparently it looks like there, if this is a red blood cell which is devoid of, it is a like kind of hollow stuff with something like the side view is concave disc, biconcave disc, if you look at the RBC. So, it looks like the extracellular composition in such that it does not get adhere at any site, which is remarkably different from an adhering cell, where the extracellular protubation which are coming out from the cells have the ability to adhere. So, if one has to develop a culture system for non-adhering cells, adhering we have talked about, they stick to a surface, and then we have to have a different kind of paradigm. Realize, and this paradigm has to be totally different. And talking about this, while citing this example of bone marrow, let's talk about the third aspect of, we have talked about oxygen and CO2, we have talked about ECM, and we have briefly talked about adhering and non-adhering cells. Now, let us talk about the concept of cell cycle. This is we are talking about under the broad heading of biology of cultured cells. So, before I move on to the cell cycle part, I wish to highlight one point which I missed out while I was talking to you about oxygen and carbon dioxide. So, if you realize, in our body the oxygen supply is met by the molecule called hemoglobin, Hb, hemoglobin, which is a complex, it is a globin protein with a heme complex. Heme complex is nothing but iron, and it's form a coordination complex, and it is entrapped in a porphyrin ring, and this whole thing is kind of placed inside a protein called globin. So, this is essentially is the color imparting one called the heme fragment, and this part is the globin protein. So, that is why it makes its hemoglobin, and hemoglobin is responsible for attaching to the oxygen and transporting it. So, hemoglobin is the key part of the RBCs, and RBC is responsible for going to, so this is how in a real life scenario happens, like these are the vessels or the veins, and these are the arteries, and of course, there is a connecting zone something like this, where the capillaries and they exchange, then there is a narrowing down which is happening like this. It's a very simplistic way, I am just putting it together, okay, something like this. Okay. So, now concerned tissues are sitting out here. The oxygenated blood or the red blood cells travel here, and unload their oxygen. This oxygen is taken up by the tissues, and the veins out here picks up the CO2 which is given away by these cells, and this is brought back via heart, goes to the lungs, and from the lungs again it picks up the oxygen, throw away the carbon dioxide, picks up the O2, throw away the CO2, and the oxygen further comes back after attaching with the hemoglobin. Now, if you realize each one of these tissues which are there in our body, they are not continuously in an oxygenated environment. So, the oxygen profile, if you just try to imagine in any part of your body, is kind of fluctuating in a manner like this. Realizing, because there is a flow which consumes, and parallelly with a small delay you will see, if this black curve what I drew is of oxygen, then there will be a carbon dioxide peaking which will be something like this. Okay. So, what essentially I wish to highlight, which I forgot in the previous class, is oxygen and CO2 is dynamically changing in a real life scenario. But if you grow cells in a dish like this, say for example, this is the dish where you are growing the cell. So, what we essentially do is that we keep the gaseous exchange, because there is no other way for us, gaseous exchange something like this. If I had to draw, it will be something like this for both the gases, or with a little bit of a, say for example, if one color represents one gas, the other one will be something like this, which is absolutely different from what happens in the real life. But then how we could get around this problem? That will introduce us to the world of the next gen cell culture technologies, which will be just like these kind of vessels, what you see, the arteries and the veins. The next gen cell culture technology will be using microfluidic systems to mimic the conditions which are there in real life, and maybe such microfluidic structures will give us a very different understanding about the dynamics of cellular growth. It is a continuous process, 24/7 multiplied by x, as long as those cells or that individual is alive. It is a continuous process which is happening. Heart is pumping, the blood is going, loaded with oxygen, downloading the oxygen at a specific, at all tissues, picks up, upload the carbon dioxide, comes back, again do the same thing in a sinusoidal manner. It is continuously varying. But could we mimic those conditions? A food for thought will be coming later. Once we will talk about this microfluidic channel and use of microfluidics, how to do all that kind of things, how people are progressing, what are the next level of technologies, and what are the technological blockages. There are so many blockages also, it is not so easy. But such systems are, unlike last 100 years, these are more dynamic system. So, you can say that modern cell culture, from static cell culture the future will be more of a dynamic cell culture, and such dynamic cell culture will be needing lot of understanding of engineering, and within engineering microfabrication, microfluidics, mass transfer, diffusion, lot of understanding of transport phenomena, lithography, and other allied techniques. And fluid mechanics. So, 100 years down the line, probably the way today's textbooks are written, those will change, because we are ushering into a very different time, where slowly we are realizing that we cannot study a system of this magnitude, where every second or every minute the milieu is getting freshened up or getting perturbed by static systems. Not only that, okay, just when I drew this, so there is always information exchange which is taking place between these multi-system systems, multi-system structures. So, say for example, one simple example, let me give you what I mean by that. So, you know that we are being always advised, and in the morning go out, and because there is UV rays which are slightly helpful. So, once we absorb the UV, what happens? Steroids derived from cholesterol, which are present in our skin, underneath the skin, they get converted into vitamin D, and for this reaction to happen, you need ultraviolet rays. Now, once that happens, the vitamin D, which is also called cholecalciferol, from your skin, just think of it, from your skin is transported to the liver. This is cholecalciferol or vitamin D. It is transported to the liver. In the liver, this cholecalciferol is converted into a intermediary analog. This intermediary analog is transported to the kidney, where this is transformed into an hormone called calcitriol, and calcitriol is a hormone which is secreted by the kidney, which is responsible for absorbing calcium and phosphorus by the bone. So, what you see here is there is a crosstalk happening between skin, liver, kidney bone. That's the complexity, or that's the integrity of such systems are. So, future of cell culture technology will be very, very dynamic. So, that is where we digressed when I told you that, whenever we will have to talk about these things, we have to talk by pulling everything together. We cannot talk in isolation that, I see this, I observe this in, but then you have to have what is the implication of it, why this particular cell type behaves in such a way, where we maintain the conditions what is needed. And again, one has to realize, if we maintain a higher oxygen tension, this may lead to oxidative damage, because higher oxygen tension always leads to the free radical formation. That is the penalty we pay for being an oxygenated environment. So, we have to ensure that the oxygen tension is not maintained at a higher level for a prolonged period of time. Similarly, that brings us to the fact that how very systematically we can keep the oxygen tension low and ensure there is not much accumulation of CO2. Having said this, this brings us to two different situations. Sometimes such situation, this situation which is currently prevailing, there is a constant level of CO2 and oxygen which is mostly supplied by the air, unlike what is happening in real life, can lead to certain cell cycle issues. So, let us first of all talk about where we were supposed to start is cell cycle. So, here let me highlight that you can, in the previous class I told you that you can distinguish between adhering cell and non-adhering cells, right. Now, I am introducing that you can classify the cells under two groups, dividing cells and non-dividing cells. Okay. So, let's put it. So, I am classifying the cells now as non-dividing and dividing. Okay. So, if we talk about dividing and non-dividing, what are the cells which comes in your mind? One of the cells which you must all be aware of are neurons. They do not divide. At least the terminally differentiated neurons do not divide. So, I use two terms here. Terminally differentiated, what does that mean? I will come to that. Okay. Similarly, there are cardiomyocytes which do not divide. Okay. Yet, you have your skin, the whole surface. There is a continuous rejuvenation, there is a continuous division which is taking place at the epidermal layer from all the way, if you remember, all the 5 layers, there is continuously a rejuvenation process which is happening. Now, at times that can become uncontrolled, as we have discussed in the previous class, which may lead to cancerous like situation or tumor. So, what we will do next is we will talk about the different stages of division and how that influences the way we are conducting the culture. Okay. So, I will close in here. In the next class, we will start off with the cell cycle. Thank you.